Sterilization. Sterilization is the destruction of all microorganisms and can be achieved by either physical or chemical methods. Sterilization is necessary for medical devices penetrating sterile body sites. Cleaning to remove visible soiling and reusable equipment should always precede sterilization. All materials must be wrapped before sterilization. Only wrapped or packed sterilized materials should be described as sterile. Before any instrument or equipment goes under the process of steam sterilization, the following should be checked. Ensure that the instrument can withstand the process. For example, steam under pressure. Ensure that the instrument has been adequately cleaned. Ensure that the instrument does not require any special treatment. Ensure that the records of sterilization process and for the traceability of the instruments are kept. Instruments and equipment will only be sterile if one of the following sterilizing processes is used: steam under pressure, that is moist heat, dry heat, ethylene oxide, peracetic acid, hydrogen peroxide plasma, and other chemical sterilant systems or sterilants, or irradiation. The above sterilizing methods are designed to give a sterility assurance level of at least 1 in a million as long as the process is validated and is according to the manufacturer's guidelines. Steam under pressure, moist heat sterilization. This is the most efficient and reliable method to achieve sterility of instruments and equipment. It is inexpensive, non-toxic, and very reliable. There are several types of steam under pressure sterilizers, also called as autoclaves: downward gravity, displacement sterilizers, jacketed and non-jacketed. These are designed for sterilization of waste, solutions, and instruments. Self-contained or benchtop sterilizers. These are recommended for office-based practice as they are able to do small quantities of fairly simple items. Pre-vacuum or porous load sterilizers. These are not suited for liquid sterilization but are optimized for sterilization of clean instruments, gowns, drapes, toweling, and other dry materials required for surgery. All autoclaves are pressure vessels. They must conform with the relevant regulations and are therefore subject to regular inspection for safety purposes. A clean, separate room is required for autoclave. Appropriate standard procedure should be followed for removing the sterilized items without contaminating while removing. All sterilized items must be placed on a clean trolley wearing PPE. Sterilization controls. There are various methods which are used to check the completeness and efficacy of sterilization process and penetration of steam inside the test load. Biological testing for sterilization. It is done by using biological indicator, Bacillus stereothermophilus.
Material required: commercially available plastic vial containing strip coated with 10 to the power 6 spores of Bacillus stereothermophilus and inner glass vial containing culture media and water bath.
Procedure: the vial is kept inside the autoclave along with the load. After completion of sterilization cycle, that is at 121 degrees Celsius for 15 minutes, the vial is brought to microbiology laboratory along with filled form, which include information about the place, time, holding period, type of load. The inner vial is crushed with the help of a hard object and it is incubated at 56 degrees Celsius for 2 days in water bath. A control vial is also crushed and incubated along with the test vial. Spores are destroyed if sterilizing conditions are made. The growth can be read manually or with the help of an autoreader. With the use of an autoreader, results can be obtained within 1 to 3 hours. Interpretation. Growth is indicated by change of color in the vial. For example, violet to yellow after incubation period. No color change indicates sterilization process by autoclave is complete and the report is sent as satisfactory, while change in color indicates improper sterilization cycle and report is sent as unsatisfactory. test is valid only when controls show color change. Turnaround time is 3 days. Bowie-Dick test. It demonstrates even and rapid penetration of steam in a steam sterilizer or autoclave. If air and non-condensable gases are not removed from the chamber and the load or from the steam supply, they may become trapped within the load. This would prevent penetration of that part of the load by steam, and consequently, sterilizing conditions may not be obtained. Materials used. Towels. Ideally, huckerback towels are used. They are thoroughly washed after purchase and before use. The towels should not be starched or ironed and should be washed once a week. The tape recommended is autoclave tape, which is normally used as a process indicator. The paper should be an A4 sheet of white wood-free copier paper. Procedure. Making the test pack. Each towel should be folded in half 3 times to provide 8 thicknesses and placed one on top of the other to form a stack of 30 cm into 22 cm into 27 cm. About 25 to 30 towels may be needed. The stack may be placed in a dressing casket or a box made of cardboard or metal which is not airtight. The stack can be wrapped in linen, paper, or secured with tape. An adhesive tape, which is printed with a chemical substance, is fixed in the shape of a cross. This test sheet is placed in the center of a stack of towels. The test pack should be placed on the longitudinal center line of the chamber 10 cm away from any door and 10 cm above the chamber base of an otherwise empty sterilizer. The towels should be lying horizontally. It must be subjected to standard sterilizing cycle. The holding or sterilizing time should not be more than 3.5 minutes at a temperature of 134 degrees Celsius to 138 degrees Celsius. Reading the test. The tape will show a uniform color change if all the air is removed and steam penetrates rapidly and completely. If all the air is not removed, then air will collect in the form of a bubble. The color of the tape in the region of the bubble will be paler than elsewhere because of the low temperature and reduced moisture level. Chemical indicators are also commercially available. Physical indicators to check sterilization process are temperature and pressure monitors. They can be in the form of graphs formed in each sterilization cycle.
Please subscribe to Medical Laboratory Scientist on YouTube. Thank you.